Everybody. Hope you're having a nice day in Vienna. Thanks to the organizers for having me. I am the founder and CEO of Accelerate Biosciences based in Philadelphia, Pennsylvania, in the US. And today we're going to be talking about our stem cell platform called a human trophoblast stem cell. And this is the earliest cells that you could possibly find without any ethical issues. Okay. So our company is based on a stem cell platform, the human trophoblast stem cell. We're based in Philadelphia, formed in 2013. It's a U.S. Delaware C Corp. We have full freedom to operate over these stem cells that we call hTSCs. And my job is really just take them to GMP, which we did last April. And now I'm providing a platform for as many people as I possibly can, including you. So at the end of the presentation, I'll have a QR code. If you scan it, I promise I'll give you the slide so you can just listen in with attention. Okay, so in regenerative medicine today, the hardest thing to do is to pick the right stem cell starting source, right? You face ethical issues, you face inconsistent cell source. You have to choose between scalability and immunogenicity. And many times there's a lot of unclear FTO or IP that patents all over the place. And because many projects come out of academia, the CMC or manufacturing is not exactly done properly to the FDA standards. And so we've gone through and actually checked off all of these boxes for everybody, right? So that we can provide a really great starting point for anybody that uses our stem cell source. Our cells have quite interesting characteristics. First, they express markers of all three germ layers. They are ethically sourced. They're scalable. They're immune privileged naturally. They're pathogen free, and they have chromosomal stability to very high populations. And we have a large IP estate to protect it. And people ask, well, who's your competition? It's actually all the other stem cell lines, iPSCs and human embryonic stem cells, HESCs or MSCs, mesenchymal stromal cells. The bottom here, you will see a continuum of fetal development and where all the different stem cells come from, right? The first 8 weeks is called the embryonic period. After 8 weeks, you have the fetal period all the way to 38 weeks. That's the birth event. That's when a baby actually is born. And anything after that is considered adult, right? So the earliest cells you can get is human embryonic stem cells, but that's usually from leftover embryos in IVF clinics, right? And then the next is ours, human trophoblast stem cells, which we get from an ectopic pregnancy at 4 to 8 weeks post-fertilization. And anything after that is fetal, but fetal are usually from aborted fetuses, so there's a lot of ethical issues with that. And then the birth event, which will get a lot of cells, usually like cord blood, Wharton's jelly, placental cells, and whatnot. And of course, you get a lot of adult cells, bone marrow, adipose, and thanks to Professor Shinya Yamanaka from Japan, they were able to take any adult cell and drive it back to a natural stem cell state called induced pluripotent stem cells, or iPSCs, right, with the 4 transcription markers. So the hESCs are the earliest, but because you get them from an IVF, every time you use them, you're technically destroying an embryo. So that's a very difficult issue. So our cells then turn out to be the earliest that you can get without any ethical issues. So on the left side here, you see a blastocyst at 5 days. At 5 days, the cells turn into two types of cells: the inner cell mass in the middle here that you could see, that later becomes the baby, the fetus and the baby. The outside ring you see is the trophectoderm, that later becomes the placenta. And in an ectopic pregnancy, what happens is the embryo is traveling down a fallopian tube and it gets stuck. And it continues to grow until it bursts the tube. And when it bursts the tube, the mother may die of internal bleeding. And so what happens is a woman will feel a sharp pain all of a sudden, not knowing that she's pregnant, go into the emergency room, get scanned. The doctor will say, sorry, you have an ectopic pregnancy. We have to schedule an emergency salpingectomy to remove it before it bursts. And the salpingectomy is where you ablate both sides of the fallopian tube and you use a scope to remove the mass. And once you remove it, all you have to do is give it to the pathologist who looks at it, confirms that it's ectopic, and throw it away. Now, why do they throw it away? The blastocyst on the left side here needs to implant into the mother by day 7 or day 8, but you don't discover an ectopic until 4 to 8 weeks after. So there are many weeks in between where that embryo is completely not connected to the mother. There's no nutrients going to it. So most OBGYNs understand that the embryo is already non-viable. Okay, so that's why when they take it out, all they have to do is confirm it with a pathologist, and then they can throw it away. Okay, so my father actually discovered this platform. He's an MD PhD, Professor Jiaonan Lee. He got his MD from Tohoku University in Sendai, Japan, and his PhD at University of London at St. Bart's. And in 2003, he was performing this salpingectomy procedure, and he took the mass out, and he was thinking to himself, well, there's got to be amazing stem cells in here. Why are we throwing it away? Right? But to avoid all the ethical issues, he decided to scrape off the preplacental tissue called a chorionic villi, which is very unique on the right side here. And he hands the mass back to the pathologist who checks it and they throw it away. And that's how we get to have the earliest stem cells without any ethical issues. Because one, you have to make this procedure to save the mother, because if the tube bursts, she may die of internal bleeding. And at the same time, the fetus is already non-viable. And so you have to save the mother, it's non-viable. And so all we're doing is harvesting the preplacental tissue of what normally would be thrown away anyways. Okay. So we have all the patents around it to protect it, and now we're making this available for everybody to use. So we're going after a couple of programs. The first one is Parkinson's disease. And there's a company called Blue Rock in the US that has already taken their program to phase 2, and they're using an hESC, human embryonic stem cell. And it takes them about 16 days to turn their cells into a neuroprogenitor cell, okay? That is Th positive, that produces dopamine. Now, our cells, we have a 24-hour procedure that can turn them into a neuroprogenitor cell that is already Th positive, right? So that it saves so much more time to be able to get to the same place. And we've done 2 animal studies already, preclinical, on this. Our second program is type 1 diabetes. And there's a company in the US called Vertex that's also gone to phase 2, but they're using an iPSC, and the induced pluripotent stem cells. And it takes them a 7-step process to get to a beta islet cell that produces insulin, right? So our cells, it takes only 24 hours to drive them to a pancreatic progenitor cell that also produces insulin. Now, a lot of people ask me, well, that's kind of strange, Utah. Why do your cells at such an early stage can produce insulin when it doesn't have a pancreas, right? Well, if you think about it, when the embryo is attached to the mother, what happens when a mother takes a big bottle of Coca-Cola, right? All that sugar going through her system. How does the embryo deal with the hyper and hypoglycemia from that bottle of Coca-Cola? Well, it turns out the placenta has a role to regulate everything coming through the blood to the embryo in order to protect it. And so it makes sense that when you put a glucose response test to placental cells, insulin may come out too. So with our cells, they do. Okay. This one is the most interesting to me, and I'm going to tell you a story. So a year and a half ago, the National Institute on Aging, which is a subsection of the National Institute of Health in the US, based in Baltimore, calls me and says, Utah, we saw one of your presentations in Seattle, and we hear that you have one of the earliest stem cells without any ethical issues. And I said, yep, that's what we have. In fact, we had already made it to GMP. And he says, can you send me all your conditioned media? I said, sure. So we sent 6 liters of frozen media to the NIA. And then I called him up and I said, hey, man, what are you doing with this? Right. And he proceeds to tell me this amazing story about a set of experiments done at Stanford 20 years ago called heterochronic parabiosis. And what they did was they stitched 2 mice together. So that would have one circulatory blood system, one old mouse and one young mouse. And it turns out that the old mouse is biologically getting younger and the young mouse is biologically getting older. Whoa, really strange. Second experiment they did was they did not stitch them together and did a plasma transfer. And the same thing was happening. So what does that mean? That means the rejuvenation of the old mouse was purely a function of the proteins coming off the blood of the young mouse, right? So if that's true, the researcher at the NIA was thinking to himself, then I wonder what would happen if you take the earliest stem cells that you can find, but without any ethical issues, because that's important for the NIH, and apply the proteins coming off of it to a senescent cell. Okay? And that's what they did. And I'll explain that data to you in a little bit after we go through the characteristics. So first characteristic, our cells are naturally senescing cells, but we are able to push them out to 85 population doublings. Now, 85 population doublings is e to the 25 cells. That's literally enough cells to create 2 trillion human beings. We have more cells than you can ever use. Compared to a non-scalable MSC, which is down here, about 25 population doublings, about 10 passages, it'll stop, right? And so that's a problem. That means every time you go back and ask for a new donor source, your use of that source is very limited. But with ours, because you can grow so many cells, scale has been solved. Okay. Also, because our cells are from a preplacental source, they all express a surface marker called HLA-G, human leukocyte antigen G. Okay, and this is the same marker that protects the embryo from the mother's immune system. Right. So think about this: the embryo is a recombination of the father's gene and the mother's gene, which is an alien thing. How come this thing can attach itself to the mother without her immune system going completely crazy and trying to reject it, right? Turns out that the placenta has a role to kind of mediate that relationship, right? With the HLA-G, it dampens down all of the mother's immune system so that it allows the embryo to stay attached. And this is the same marker that now allows you to be able to plant an embryo into a surrogate mother, somebody who is biologically completely unrelated, to be able to carry the baby for you to term, which is a really strange shortcut that we discovered, right? But nevertheless, very amazing. Now, when we differentiate our cells into a neuroprogenitor cell or the pancreatic progenitor cell, the HLA-G sticks. That means that these cells are really ideal for allogeneic therapeutics. And in fact, we've taken these human cells and we put them in 5 different animal studies into immune competent Sprague-Dawley rats. So human cells and fully immune competent rats. And we see these human cells survive and they home and regenerate the affected organ. We don't see any teratoma or tumor formation, and we get all behavioral improvements. Pretty amazing. So we're the only stem cell platform that you can find today that has scale and immune privilege at the same time. Now, when we did the population doubling, we scaled it out to 85. The last karyostat that we did was at 72 population doublings. And the karyostat tests for genetic stability. And as you can see here, very, very, very stable genome. And we believe that the reason is because these cells have literally been incubated in a fallopian tube. And they've never gone into the endometrium or the uterus or implanted and never gone through the shocks, the epigenetic shocks of becoming a human being or a baby, right? And so these cells are very, very early on, and we extracted directly from the fallopian tube. That's why their DNA is very, very stable. We currently have 16 different, that's 16 different GMP compliant donor lines already banked away. Okay. And we get them from Taiwan. And in Taiwan, quite unfortunately, a third of the women have HBV or hepatitis B, and more than 90% have CMV. Okay. Now, when we discover they have HBV, we have to throw all the cells away according to the FDA rules. But what I did was I took all these samples and I sent them to IDEXX in the US to test for H-impact 1, to test for 25 human pathogens, and they come back completely clean. That means, so even though the mother has HBV or hepatitis B or CMV, the cells do not. Okay, so there's a defensive mechanism at the fetal maternal interface that we don't fully understand because we can't do any research on healthy pregnancies, right? But now what we know, that once the HPV, we have to throw all the cells away. But at least we know that the cells that we do have are completely clean and probably the cleanest starting stem cell source that you can find. We've also expanded our cells. These are naturally adherent cells, but we've expanded it in 3D with microcarriers to a 5-liter scale, and we're currently harvesting all the exosomes from our hTSCs. And this is in collaboration also with the NIA. We in-license an mRNA technique to drive our hTSCs to an iPSC, just like the Japanese, okay? And why did we do that? Well, we decided that because we discovered that the hESC, the human embryonic stem cell, and the iPSC protocols do not work with our hTSCs. So we had to discover our own protocols, but that would take too long. So we wanted to provide a platform where everybody can easily adapt and use publicly available protocols. So we created an iPSC platform for everybody to use. And as a proof of concept, we took the iPSC, we took a kit from a public source and drove them to an NK cell. And they kill like gangbusters, right? So they kill a lot. And a couple of things I want to note here. So typically, when you take a cell from a donor, it's like fibroblast or cord blood or an adult cell, and you reprogram to an iPSC, there's still a consenting adult walking around in the world. Okay, technically with the same DNA as the cells that you're using. And it causes a little commercial risk because a few years back, I don't know if you may have heard, Thermo Fisher was sued by the Gila estate, Henrietta Lacks estate. And Henrietta Lacks was a woman that stepped into Baltimore in the 1950s with cancer. And one of the professors or doctors took her tumor sample and discovered that the tumor was growing infinitely, right? It was immortal. And they kind of bottled it up and started using it for cancer research. And so the family came back and sued Thermo Fisher because they never got consent from the woman. And so they settled out of court. So now, at least for commercial use, there's a very big risk. Okay. Second thing is typically when you convert fibroblasts or other types of cells into an iPSC, the conversion rate's around 15%. But with our cells, they're like 70%. And we believe that's only because our cells are super, super early and they're easy to convert. And also the third thing is with our cells, when you convert them to an NK cell, they naturally kill. They don't have to have an extra activation step. With other cells, you may have to do that. Okay. So our iPSCs are quite special and unique. We also have a landing pad called GEMS that our CSO created. And that means that you just have very specific parking lots where when you use CRISPR or zinc finger or TALEN's gene editing tools, you can insert a gene or a CAR into that one specific spot and then it won't go anywhere else. We've effectively solved the random insertion and off-target problem, right? And so right now what we're doing is creating a plug-and-play iPSC system. And now imagine an iPSC with this platform. Any immuno-oncology company can now come to us and say, hey, Utah, we'd like to try, say, CD47 on pancreatic cancer. We'd be able to put that CD47 plasmid directly into one of the parking spots, then take the whole iPSC and differentiate to all the various effector cells, T cell, NK, macrophage, etc. Then be able to use that effector cell on the actual target and be able to tell that company, hey, listen, for your target and your CAR macrophage is probably the most efficient. So we can help them de-risk it. And we're working with Mahendra Rao, one of the former director of the Regenerative Medicine Institute at the NIH on this program. All right. Lastly, we're going to go through some of the data. for the NIA work that we did. So we're working on cell senescence. And so the National Institute on Aging is focused on cell senescence. And cell senescence is one of the 12 hallmarks of aging. These are all the reasons why we age, what causes us to age, okay? And a senescent cell is just cells that are super old and they don't divide anymore, but they still refuse to die and they stay in our bodies, right? And these cells are putting out a lot of pro-inflammatory messages, and that's linked to a lot of the disease that we have today. So the NIA is thinking, all right, how do we either kill them, called senolytics, or how do we control the messages that they're putting out for inflammation, called senomorphic? Okay. And so basically, just to repeat, they said, if heterochronic peribiosis works, what would happen if you take the earliest stem cells and put it towards senescent cells? And so we did a series of pilot studies using the entire conditioned media as well as isolated extracellular vesicles, or secretome, right? And basically in the pilot studies, we see that in both cases, we see the suppression of pro-inflammatory cytokines in the mRNA. And that was enough for us to go into a full study, characterization in vitro and in vivo. And as a short, this is the transcriptomics. We see that all of the SASPs, these are the pro-inflammatory markers from senescent cells, are completely downregulated. We also see a systemic downregulation in the proteome. So we did O-link for this. And so all the inflammatory markers are systemically downregulated. Pretty incredible. And we discovered that the reason is because our conditioned media was suppressing NF-kappa B, one of the activators of inflammation. And we were also providing defense against double-stranded DNA breaks, and that's the gamma H2AX marker. So given that, the NIH went through an N=20 animal study, and we discovered that in the plasma of the mice, the aged mice, all of the inflammatory markers were also significantly downregulated. So what we did was we went through and took 4 of the key markers, these 4, GDF15, CXCL1, DPP4, and BAFF, and we linked them to all of the potential indications on the right that you see. So cytokine storm, a lot of autoimmune diseases like rheumatoid arthritis, inflammatory bowel disease, lupus, diabetes, atherosclerosis, etc. So we're now going through and we are trying to figure out which one is the most efficient indication for us to use the exosomes in a clinical trial. So we have more than 120 patents today, with 58 already granted, and we're working with one of the top firms in the US called Wilson Sonsini, and we have full freedom to operate because every couple of years we'd file more patents, and my partner at Wilson Sonsini happens to be a head of the Boston office, and he says, and used to be a USPTO and examiner himself. So he says that the reason why is because, every time we file a new patent, the only prior art that they find are our own. So no one's coming into our field, which is great. Okay, so we have a lot of clearance. So if you work with us as a starting stem cell source, you would have the protection of a lot of patents. Okay, here's the team. I went to Berkeley undergraduate. Then I did my EMBA at China Europe Business International Business School in Shanghai, China. My CFO, Henry Chen, did his undergraduate at UPenn and EMBA at NYU. Stiko Patma, our CSO, was working on the early CAR T program in Carl June's lab at UPenn. Then he left and went to J&J and was there for 13 years and took an ocular therapy from 0 to phase 2. On the bottom here, you see my father, MD, PhD. He discovered a platform. We're working with Mahendra Rao, MD, PhD, former director of the Regenerative Medicine Institute at the NIH on the plug-and-play iPSC program. Benjamin Fryer is our CRO in the middle here. He runs Pluristics in Seattle and also ex-J&J, and he did all the work here. So everything's been validated by a third party with the exception of the animal studies, of course. And we have Aubrey de Grey. If you are in longevity or the aging field, you would definitely recognize him. And he's helping us with our work with the NIA. And Bram Bout from Holland, famously at Crucell, invented the Percy 6 line, which is a manufacturing cell line, a producer cell line. And this was sold to J&J in 2011 for $2.4 billion. And so J&J uses the Percy 6 line to make all their vaccines today. And so he is now helping me, or helping us, take an hTSC, immortalizing it, and also creating a line to hopefully replace the HEC293s and Percy 6. So we have 3 different platforms on offer: an hTSC, which already took all the way to GMP, but we're offering the research lines from the same donor line as the one we took to GMP. That means if you take the research line from us and work with it, when you succeed, all you have to do is come back and get the GMP line, and you're off and running into towards your IND. We have our iPSC, if that's what you want to start with. We have 3 clonal lines ready for you, also under RUO. And we have our exosomes, which is purified and mass-produced already. Okay. So we have perfect product market fit. We have the earliest embryonic stage cells without any ethical issues. We have a platform that has scale and immune privilege. We have the entire IP portfolio locked up with Wilson Sonsini, and we took everything to GMP already, and we're filing a DMF, a drug master file, with the FDA with the help of Dark Horse Consulting. And my job really is to speak to as many of you as humanly possible and get the cell platform to you so that you can accelerate your programs. Okay. That's it. That's my presentation. So if you scan either my WhatsApp or LinkedIn, send me a message and just say that you saw me at this conference. I'd be happy to send you a PDF of the slides. Wonderful. That's it. So does anybody have any questions?